We're on the cutting edge of research activities in cancer. Cancer is a big problem in our country. It's the second leading killer of adults in our country, and we need to understand how to treat that better so that we can save a lot of lives. So we try to understand what the cell is doing, how the cancer cell is different than the normal cell. And a cancer cell is really a cell that just has lost controls, and it divides in an uncontrolled manner. And because it divides in an uncontrolled manner, it spreads throughout the body, it displaces normal cells, and kills the person. We have 83 scientists in our center, and they are from 15 different departments around campus. Mark Hall is a chemist, and he studies specific proteins, and in order to be able to study those proteins, he tries to study very simple models. So he uses yeast.
The yeast that we study is baker's yeast, and it's the yeast that's used in the bakery to make bread. It's also used in the process of making beer and wine.
This device here is called a fermenter. This is one of the things that we use just to grow yeast cells. It allows us to grow large volumes. We've got several gallons of yeast growing here.
A lot of the cellular processes that can cause cancer in humans when defective also exist in yeast, and we can study those basic processes in yeast and then by inference understand how they can contribute to cancer in humans. These are some pictures that we've taken with our fluorescence microscope here. This picture up here is just a light microscope image of what a yeast cell looks like that's undergoing division right now. So this is the mother cell, for example, here, and it's now dividing into two cells. My interest really is in understanding how cells work and function, but that information then is very useful to other scientists who are more interested in developing ways to treat and cure and diagnose diseases like cancer.
Now we're inside the Cancer Center building itself, and we're in the mass spectrometry facility. Behind me is an instrument called a mass spectrometer. One of the things that we can do is to compare the molecules inside a cancer cell to the molecules that are inside a normal cell to try to understand what's different about a cancer cell.
Susan Mandrisa is working on brain tumors and trying to understand what makes them a tumor versus a normal brain cell.
My lab is studying a type of brain cancer that's called medulloblastoma. This is a type of tumor that forms in the cerebellum, which is a part of the brain towards the back of your brain. And this particular type of tumor is very commonly observed in small children. So one of the most frequent ways that children are initially potentially diagnosed with this type of tumor is that they start to experience difficulties with their motor coordination. So they may be less stable, they maybe fall more frequently. We've been taking the approach of really trying to use mice as a model organism to study the different signaling pathways and the molecules that are important for the cerebellum. The normal brain, which has a lot of MDM2, is larger than the mutant brain. We try to understand how the cerebellum is developing in order to try to understand how this particular type of pediatric or childhood tumor of the cerebellum also develops. There are kids out there who need the results of this research.
It's interesting to learn new things about life, but it makes it more satisfying when you know that information is going to be useful to help society.
You are making discoveries. You are discovering things that yesterday no one knew.
When we can take those discoveries and develop new treatments, and we see patients who are responding well and living longer and sometimes cured, that's the most gratifying thing that exists.